So, we are back doing more revision. Today's stream, this is a bit off. Today's stream, we are going to be looking at lipids. I was going to do proteins and enzymes, probably saving that for tomorrow. And then once we're done with proteins and enzymes, we'll do a big stream on Friday going over DNA, RNA, transcription and translation. And we probably don't need to cover ions or water very much. So let's just move right on to lipids, because lipids is a relatively simple topic. I like to think I have a good knowledge on it. So let's just see what we can remember. See if we need to work on anything real quick. Where's my red pen gone? Okay. So, lipids. So, the first thing you need to know about lipids is the type of bond, which is an ester bond. Okay? So, in proteins, the bonds that join the amino acids together are called peptide bonds. In carbohydrates, the bond that joins them together is glycosidic bonds. The bonds are ester bonds in lipids. So, the first lipid I will draw is the phospholipid, which I'm pretty sure resembles something like this. In other words, there are two structures, or two parts of this that you need to know. Okay, two parts. The first part is the chains. Second part is the head. Now the head, and this is very important, is hydrophilic. Meanwhile, the chains are hydrophobic. Now, these have a charge, but I don't recall which way around it is. This is a phospholipid, by the way, so I need to add one more thing to it. But I'm just going to cover this bit first. I don't recall whether it's positive on the head. Chains... it might be positive. Let's just say for now, because again, this is just revision. I'm hydrophobic. You are not hydrophobic, buddy. You're a different kind of phobic. Obviously we're forgetting here the phosphate group, which is just a P. We'll just put it as a P for phosphate. So, phospholipids form a phospholipid bilayer, obviously, in the cell surface membrane. They form a bilayer. And it's actually quite a prominent exam question that you could get asked, where they will ask you, why does it form a bilayer? And you basically just need to talk about the interactions between the hydrophilic head, hydrophobic chains, which causes them to double up, basically, because otherwise this is going to be touching the extracellular liquid. So instead of that, that's touching the extracellular liquid. So they're in their conditions that they want to be in. So that is the first type of lipid. But the main one we will cover now is the, I think it's triglyceride. I believe that's its name. Triglyceride. Triglycerides are chill as hell, really. Pretty sure that the whole thing here is... it might be hydrophobic. I don't recall. There is something else I need to cover, but I will go on to that in a sec, about the chains and the unsaturated and saturated. We'll get onto that in a sec. Triglycerides, you look at a lot in digestion, because the idea with triglyceride is that they become broken up into their component molecules. Then they're put in vesicles. Vesicles form chyromicrons, etc. So I think that's everything for triglyceride. Now, let's talk about the chains. There are different types of chains. Unsaturated fatty acids and saturated. Saturated and unsat. The difference between saturated and unsaturated is simple. In saturated fatty acids, they are saturated full of hydrogen. So carbon can bond to 4 molecules. So we're going to have a chain that looks like this. Cool. Meanwhile, unsaturated have double bonds. So that carbon is bonded 4 times. It can't have another bond with hydrogen. This one can, because it's only bonded there. And that one can, yeah. But the others can't. So the difference between saturated and unsaturated is unsaturated have double bonds, saturated have single bonds. Saturated contain the maximum amount of hydrogen. Unsaturated contain the minimum amount of hydrogen. I will check the definitions later to make sure I haven't missed anything, because I know they can ask you this question in an exam as well, and you could be prepared to get that wrong. Thank you. Unsaturated. So, that is a general overview of lipids. The interesting thing to note is this one is going to be unsaturated, this one's saturated, because this has a chain that kinks slightly. It's slightly bent to the side. All of these are straight, which means that they're saturated. Monoglyceride, diglyceride, etc. Just in case you didn't get the idea there. Trying to think what else there is to do with lipids that we need to cover, because honestly, this topic is really short. We have the test for lipids, the emulsion test. Emulsion is a suspension of one solution in another, I'm pretty sure. It's a definition like that, a suspension of a solution in another solution, alright? A suspension of solid in a solution. Something like that. Just play around with those words until it sounds right, but I will look for a proper definition. Again, it's been a while since I've done this topic. The way we would test for something, first of all, would be using a pestle or mortar, grinding up our food, or our whatever we're testing, putting it in a test tube, or a boiling tube, I guess. You would add ethanol. And I believe that would work. So you would grind it up, add ethanol, shake it, and if it forms a cloudy white substance, that is an emulsion. I think you're not allowed to mention milky white. You won't get marks if you say milky white, but you will get marks if you say cloudy white. So cloudy white suspension, that is emulsion. Or cloudy white emulsion is a suspension. So yeah, you would, break apart the food with a pestle or just whatever. Add it to the thing, add ethanol, shake. And I think that's about it. I do not recall much else about lipids, to be completely honest. I'm trying to think if there's anything else we've missed. We could quickly talk about lipid digestion, but it has been a while, and I don't remember much of that. So I think we should save that for another day when I'm able to absorb more information, because I'll be able to nail the definitions then. All I remember is you have bile, which... Does it emulsify the fats? And then the bile salts associate with the broken apart glycerides, goes through there, forms a... Yeah, I'll remember it some other time. But I think that's about it for lipids. I don't think there's much else that I really need to go over there. The plan is, once we get the biological molecules topic finished by at least Saturday, I'll be ready to move on to, I think we'll go right on to cells. We'll look over mitosis, we'll look over meiosis, then we will go on to transport across membranes, which is a really nice, easy one if you remember the definitions. We'll look at, cell components of a cell membrane, the fluid mosaic. And by the end of this month, by the end of November, should be hopefully finish every topic. We should be looking at mass transport, things like the insects, fish, ways of gas exchange, things like hemoglobin, the components of that. We could look back at the genetic topic again, looking at things like the difference between prokaryote and eukaryote DNA. Honestly, there's a lot for us to cover, but it's more of a choice of which one do we do first and which one's the most important. So I think biological molecules, while it is important, it's probably one of the easier topics for me to remember, especially since we've done ATP revision. So I think the plan is now, I'll probably wrap up this stream now, and next time I stream, we'll be looking at proteins and enzymes. So take care.